A warm greetings to all viewers. Today we are going to learn about the patterns and phases of development along with cell-cell interactions. So first, it is mandatory to collect some historical perspective and the basic concepts that lay behind the development. History and basic concepts. Developmental biology is a fundamental aspect of biology. Development depends upon complex mechanisms and many layers of biological information that are superimposed one upon another. As our techniques improve, developmental biology has the potential to be much better understood. Once, developmental biology was mostly descriptive, that is, descriptive or comparative embryology. Modern developmental biology is mostly experimental. Recent advances in cell biology, genetics, and molecular biology has and will continue to further our understanding of developmental unlike any time in the past. Embryogenesis, that is, embryo formation, determines the overall body plan. Organogenesis, that is, organ formation, determines subsections of the body, for example, vertebral limb, Drosophila eye, etc. Often these processes share much more than is first obvious. Many genes, proteins, signal transduction pathways and cell behaviors are common to both processes. Epigenesis versus pre-formation. Epigenesis, that is, upon formation, is a theory of development that states that new structures arise by progressing through a number of different stages. Pre-formation theory suggests that all structures exist from very beginning, they just get larger. One pre-formation theory, the theory of the homunculus, suggested that a little human embryo was hidden in the head of every sperm. Since an embryo grows to be an adult and that adult produces another embryo and so on indefinitely according to the theory of preformation, the very first embryo must include within itself tiny copies of all the future embryos. Cell theory. Organisms are composed of cells, the basic unit of life. Both animals and plants are multicellular composites that arise from a single cell. Therefore, development must be epigenetic and not preformational, since a single cell, that is the fertilized egg, results in many different types of cells. Only the germ cell, that is egg and a sperm, pass characteristics to the offspring. Somatic cells are not directly involved in passing on traits to the next generation, and characteristics acquired during an animal's life are not passed on to the offsprings. Remember that a hen is only an egg's way of making another egg. Meiosis and fertilization. Meiosis is the reduction division that allows diploid precursor cells to generate haploid germ cells. At fertilization, a diploid is reformed by joining two haploid germ cells. The diploid zygote contains equal number of chromosomes from each of the two parents. Observations of Siachen eggs revealed that after fertilization, the egg contains two nuclei which fuse to form a single nuclei. The nucleus must then contain the physical basis of heredity. Genetics has been key to development. The developmental genetics of Drosophila melanogaster and Caenorhabditis elegans are the best known examples. Homologous genes identified in these organisms are found in other species. Dominant or semi-dominant mutations have one copy of mutant gene produces called mutant state. Recessive mutations are two copies of a mutant gene gives the mutant state. Mutants can arise spontaneously, but induced mutation and screening has become the standard way of identifying developmentally important genes. Mosaic versus regulative modes of development. Mosaic development depends upon specific determinants in the one-cell zygote that are not divided equally between the daughter cells, that is, as seen in asymmetric division. Rocks in 1880s destroyed one cell of a two-celled embryo with a hot pin to result in a half of a frog embryo. Regulative development depends upon interactions between parts of the developing embryo can result in causing different tissues to form even if parts of the original embryo are removed. Drist destroyed one cell of a sea urchin embryo at the two-cell stage and a normal appearing but smaller sea urchin larvae resulted. Regulatory development induction. Induction is a type of regulatory development. This is a process where one tissue directs the development of another tissue. A classical experiment by Spearman and Mangold in 1924 showed graft of the blastopore lip of one newt onto another. Processes of development includes cleavage division with no increase in cell mass, pattern formation that includes anterior and proximal and dorsal or a ventral axis forming coordinate system, morphogenesis take 3D form, for example seen in neural crest, migrates far, cell differentiation where cells become structurally and functionally different, and lastly growth where cell multiplication is observed with increase in cell size and deposit extracellular material, for example bone, shell. Growth can be morphogenetic. Cell behaviors. Cell behavior is the key link between gene expression and development. In brief, it is gene activity that gives cell identity. It may be cell-cell communication, cell shape changes, cell movement, cell proliferation, or even cell death as seen in apoptosis. Inductive interaction. Inductive interaction is the process by which one group of cells change the fate of another group of cells. The information to cause induction passes from cell to cell in the form of secreted diffusible molecule, surface molecule receptor, and last gap junction, also referred to as channel. Competence is the state of being able to respond to inductive signals due to the presence of receptor or transcription factors. Positional information directs pattern formation. Positional information directs pattern formation by giving positional values to cells. The French flag model, blue, white and red strips, refers to the assignment of positional values in response to a morphogenetic gradient. This biological information must first be specified and then the value must then be interpreted. Morphogen varies in concentration and directs different fates at different concentrations. Development is progressive generative program. Development depends upon a progressive series of instructions. An embryo needs to have each action to be built upon the previous action and that on the one before. Development instructions are not a blueprint but is a structural list of actions. Lateral inhibition. Many structures are regularly spaced. Cells that form a structure stop neighboring cells from doing the same, as in feathers, compound eye, faucets. Lineage dependent fate. Cytoplasmic localization and asymmetric cell division control the fate of resulting cells. Stem cells can produce daughter cells that are also stem cells. However, some daughter cells become different and give rise to different lineages. Cell-cell interactions. Cells signal one another with chemicals. Receptor proteins embedded in the plasma membrane change shape when they bind specific signal molecules, triggering a chain of events within the cell. Cell signaling can occur between adjacent cells although chemical signals called hormones act over long distances. Proteins in the cell and on its surface receive signal from other cells. Some receptors are located within the cell cytoplasm. These receptors respond to lipid-soluble signals such as steroid hormones. Many cell-to-cell signals are water-soluble and cannot penetrate membranes. Instead, the signals are received by transmembrane proteins protruding out from the cell surface. Cell surface receptors often use second messengers to transmit a signal to the cytoplasm. Surface receptors and second messengers amplify signals as they travel into the cell, often toward the cell nucleus. Cells possess on their surfaces a variety of tissue-specific identity markers that identify both the tissue and the individual. Cells attach themselves to one another with protein links. Some of the links are very strong, others more transient. Adjacent cells form a sheet when connected by tight junctions, and molecules are encouraged to flow through the cells, not between them. The cytoskeleton of a cell is connected by an anchoring junction to the cytoskeleton of another cell or to the extracellular matrix. Many adjacent cells have direct passages that link their cytoplasms, permitting the passage of ions and small molecules. Pattern formation. Pattern formation is the developmental process by which cells acquire different identities depending on their relative spatial positions within the embryo. Pattern formation ensures that tissues and organs develop in the correct place and orientation within the body. The science of pattern formation deals with the visible, statistically orderly outcomes of self-organization and the common principles behind similar patterns in nature. In developmental biology, pattern formation refers to the regeneration of complex organizations of cell fates in space and time. Pattern formation is controlled by genes. The role of genes in pattern formation is an aspect of morphogenesis, the creation of diverse anatomies from similar genes, now being explored in the science of evolutionary developmental biology or EVO-DEVO. The mechanisms involved are well seen in the anterior-posterior patterning of embryos from the model organism Drosophila melanogaster, a fruit fly, one of the first organisms to have its morphogenesis studied, and in the eye spots of butterflies, whose development is a variant of the standard fruit fly mechanisms. Let us learn with an example of anterior-posterior axis patterning in Drosophila. One of the best understood example of pattern formation is the patterning along the future head to tail, that is anterior to posterior axis of the fruit fly Drosophila melanogaster. The development of this fly is particularly well studied, and it is the representative of a major class of animals, the insects. Other multicellular organisms sometimes use similar mechanisms for axis formation, although signal transfer between the earliest cells of many developing organisms is often more important than in Drosophila. There are three fundamental types of genes that give way to the developmental structure of the fly: maternal effect genes, segmentation genes, and homeotic genes. The development of Drosophila is particularly well studied, and it is representative of a major class of animals, the insects or insecta. Other multicellular organisms sometimes use similar mechanisms for axis formation, although the relative importance of signal transfer between the earliest cells of many developing organisms is greater than in the example described here. Formation of dorsoventral axis. Formation of the dorsal ventral axis is dependent on the ventral nuclear concentration of a maternally synthesized transcription factor called dorsal. The determination of the dorsal site of the embryo occurs during oogenesis, when the oocyte nucleus moves along microtubules from the posterior to the anterior dorsal margin of the oocyte. The nucleus expresses a protein called gherkin, which is secreted locally and thus only activates follicle cells in the dorsal region by interacting with the torpedo receptor. This inhibits the production of pipe protein, and thus follicular cells expressing pipe are on the ventral side. Pipe activates an extracellular protease cascade in the perivitelline space between the follicle cells and the egg, which results in the cleavage and the Toll ligand spatzel and activation of the Toll signaling cascade on the ventral side. Dorsal protein is present throughout embryonic cytoplasm, but bound to cactus, which prevents it from translocating to the nucleus. Toll signaling results in the degradation of cactus, which allows dorsal to enter the nuclei on the ventral side of the blastoderm. Overall, a difference in the localization of the oocyte nucleus becomes a difference in the signal state of the surrounding follicle cells, which then signal to the resulting blastoderm nuclei. Once in the nucleus, dorsal activates different genes depending upon its nuclear concentration. This process sets up a gradient between ventral and dorsal side of the blastoderm embryo, with the repression or induction of dorsal target genes being differentially regulated. At the ventral end of the embryo, blastoderm nuclei exposed to high concentrations of dorsal protein induce the transcription of the transcription factor twist and snail, while repressing zirconalt and decapentaplegic. This results in the formation of the mesoderm. In the lateral region of the embryo, low nuclear concentrations of dorsal lead to the expression of rhomboid, which identifies future neuroectoderm. More dorsally active DPP signaling represses rhomboid, thus confining it to the lateral blastoderm nuclei. At the dorsal side of the embryo, blastoderm nuclei, where this is little or no nuclear dorsal protein, expresses zirconalt, tolloid and decapentaplegic. This leads to the specification of non-neural ectoderm and later in the blastula stage of amnioserosa. The ventral activity of the TGF beta family signaling protein DPP is maintained by the expression of the secreted DPP agonist SOG, short gastrulation, in the neuroectoderm. SOG binds to and prevents DPP from diffusing to the ventral side of the embryo, and through the cleavage of SOG by tolloid also enables a sharpening of the DPP gradient on the dorsal side. The DV axis of Drosophila is due to the interactions of two gradients, a ventral concentration of nuclear dorsal and a dorsal concentration of DPP activity. Mechanisms of pattern formation in development and evolution. Evolution of metazoan organisms has produced over hundreds of millions of years both phenotypic complexities and the developmental mechanisms by which such complexity is generated, both architecturally complex and functionally coherent. How this distribution of cellular phenotypes, cell states, is attained through spatiotemporal regulation of gene interactions and cell behaviors is one of the main questions of developmental biology. To this end, considerable knowledge has been acquired during the last few decades about the genetic composition of multicellular organisms, how various genes and gene products interact, where are they expressed, and in which developmental processes are they involved. Organismal development is enabled by developmental mechanisms. A developmental mechanism is understood in this paper as gene product interactions and changes in cellular behaviors such as mitosis, apoptosis, secretion of molecular signals, cellular adhesion, differentiation, and so forth that are required for and cause the formation of a particular arrangement of cell states in three-dimensional space, that is, a pattern. We reserve the word form for the spatial arrangement of cells without considering their state. In formal terms, the development of an organism can be described as transformation from one set of pattern to another set of patterns, and here we aim to highlight the basic logic of the developmental mechanisms underlying these pattern transformations. Casual explanations of pattern formation in an embryo primordium require knowledge of all the genes, epigenetic determinants, that is, surrounding cell arrangements and other microenvironmental conditions in the embryo, and their interactions necessary for generating such a pattern from a previous pattern. In practice, causality can be inferred by testing how well a developmental mechanism predicts the variational properties, the range of potential morphological outcomes. It is common in theoretical discussions of development to distinguish two components of pattern formation. First, pattern formation through cell-cell signaling mechanisms establishes cells with different states and different spatial relationships by signaling in 2 or 3 dimensions in developing planar and solid tissues respectively. Second, mechanisms that uses cell behaviors other than signaling act on the previous established pattern to cause the formation of 3-dimensional tissues and organs. As described in detail below, morphogenetic mechanisms change the spatial distribution of cells without changing cell states. Morphogenetic and inductive mechanisms act at all stages of development. Inductive mechanisms are generally implicated in developmental changes that produce new patterns. Because induction is a prerequisite for development to proceed, no particular attention is normally paid to the order in which inductive and morphogenetic mechanisms function. We suggest, however, that the relative timing, including possible coincidence of inductive and morphogenetic mechanisms, can have major consequences for developmental dynamics and the range of potential morphological outcomes, and is therefore a central importance for the understanding of both development and morphological evolution. In fact, the terms pattern, pattern formation, and morphogenesis are often used in different and not always explicit ways. In this article, we define these terms in a way that does not make assumptions about how inductive and morphogenetic mechanisms interrelate in producing developmental change. A key aspect of our treatment is the introduction, or rather appropriation, of the term morphodynamic, distinguished from morphostatic, to characterize complex developmental mechanisms in which inductive and morphogenetic mechanisms interact with one another in a reciprocal fashion. The need for new concepts to bring order of complexities of morphogenesis were anticipated by earlier investigators such as the cell biologist Paul Weiss, whose influential textbook of developmental biology was first published under the German title Morphodynamic and later published in English as Principles of Development. The mathematician Rein Thom used the allied phrase dynamics of forms in his topological treatment of embryogenesis and human biology, structural stability and morphogenesis. In the following sections, we briefly review and classify the main types of developmental mechanisms for which there is experimental evidence. As noted, these can be characterized as basic mechanisms that employ only one or few cell behaviors. Although our main objective is to explore the ramification of the heretofore overlooked morphostatic or morphodynamic distinction. We also emphasize that efforts to formulate useful computational models of development and evolutionary developmental scenarios will benefit from an accurate schematization of the full range of experimental confirmed developmental mechanisms. Summary: we have studied that a classification of developmental mechanisms shows experimentally to generate pattern and form in metazoan organisms. Such mechanisms can be organized into three basic categories, and that two of which may act as composite mechanisms in two different ways. The simplest categories are cell autonomous mechanisms, in which cells enter into specific arrangement patterns without interacting, inductive mechanisms in which cell communication leads to changes in pattern by reciprocal or hierarchical alteration of cell phenotypes, states, and morphogenetic mechanisms in which pattern changes by means of cell interactions that do not change cell states. The later two types of mechanism can be combined either morphostatically, in which case inductive mechanisms act first, followed by the morphogenetic mechanism, or morphodynamically, in which case both types of mechanisms interact continuously to modify each other's dynamics. We propose that this previously unexplored distinction in the operation of composite developmental mechanisms provides insight into the dynamics of many developmental processes, in particular morphostatic and morphodynamic mechanisms respond to small changes in their genetic and microenvironmental components in dramatically different ways. We suggest that these differences in variational properties lead to morphostatic and morphodynamic mechanisms being expressed to different extents in early and late stages of development, and to their contributing in distinct ways to morphological transitions in evolution. Thanks.